Hey Mustangs, in this video we're going to be taking a look at what you need to know for your DNA one-on-one quiz. So first thing you have to remember is you're going to be sharing your screen with me and you're going to be using your cursor to point to and identify the key elements of the DNA structure. Up to this point, what you should have learned when it comes to DNA is one, its function. So the fact that it is a set of instructions that say how to build and then maintain a living thing. And the second thing that we focused on has been the structure itself. And that's what this quiz is all about. If you understand DNA structure, you will get just about everything that comes after this. You'll understand protein synthesis better. You'll understand chromosomes. You'll understand mitosis, meiosis, the cell cycle. Everything else will make so much more sense when you understand the structure of DNA. Now, big thing about structure of DNA, we know DNA stands for deoxyribonucleic acid. So we know it's a macromolecule, which means it's made by connecting smaller monomers together to build it. In this case, awesome thing about this is nucleic acid. The monomer sounds like it, nucleotide, so it sounds the same. So it's made out of nucleotide, connecting nucleotides together. So let's go ahead and take a look. I'm going to go over how I will ask the questions and how you should point to and identify the different things. So first question will be, since DNA is made out of nucleotides, that's one of the most important things to understand right there. I will ask you about the nucleotide. So I will say, point to a nucleotide and name its 3 parts. Now, when I look here, I can actually see my 2 strands before we even start. So I see my 2 strands here. And when I look at this strand, there's 1 nucleotide, here's 2, here's 3 nucleotides, 4, 5, 6. So I see 6 nucleotides on this side. These are upside down, but we can still identify the nucleotides. There's 1 here, 2, 3, 4, 5, 6. So 6 on this side, 12 nucleotides that are making this DNA molecule I see here. So this whole thing is 1 DNA molecule made out of 2 strands, and each strand is made out of the nucleotides. So point to a nucleotide and name its 3 parts. I'm going to get fancy and go for an upside down one. So I'm going to say, okay, here's a nucleotide. There's a phosphate connected to deoxyribose sugar, connected to a nitrogenous base. Point to and name the 4 bases: adenine, thymine, cytosine, and guanine. What are the base pairing rules for DNA? Adenine always pairs with thymine. Cytosine always pairs with guanine. How many bonds form between them? Adenine and thymine form 2 bonds. Cytosine and guanine form 3 bonds. How many strands make up DNA? 2. Here's 1 strand over here. Here's the second strand over here. So 2 strands make up DNA. What kind of bonds hold those 2 strands together down the middle? So our model helps us with this. It looks like the letter H. If I draw a line here, and then a line across, forms the letter H. So hydrogen bonds hold the 2 strands together in the middle. Point to the sugar phosphate backbones. There's 2 sugar phosphate backbones. It's the repeating orange and purple pattern we see here, sugar phosphate over and over again. So that's 1 backbone. Here's the other sugar phosphate backbone over here. So 2. What kind of bonds hold that together? C for covalent bonds. So again, our model shows us the letter C, covalent bonds, the stronger bonds hold it together. We will learn later on why we use weak bonds in the middle. Okay. All right, point to the 5' and 3' ends of each strand. So you have a 5' here, wherever the phosphate's hanging off, 3' down here, 5' here, 3' there. Next, what does anti-parallel mean? It means that the 2 strands are side by side, but one is upside down, or you could say running in the opposite direction. If you want to get really fancy, you can use the primes. So you could say 1 strand goes 5' to 3', the other goes 3' to 5'. So you could use the primes in that case. Then I will ask you, what is the true shape of DNA called? So that true shape, here it looks like a ladder in our model, but remember we have that spiral staircase or twisted ladder, that is a double helix. So the true shape of DNA is a double helix. After I've asked you those basic ones, then I will ask you about DNA replication. Okay, so you'll have it set up, you'll have your model there, you'll have all the pieces ready to go, and you have to explain to me DNA replication. Now there's a secret question. I call this secret question because it's not listed in the blue boxes here, but the secret question is, when does DNA replication occur? So we talked about this when we did our notes. We said that we never want extra pieces of DNA inside of a cell. Extra DNA is a bad thing. It's not going to give you superpowers or anything. So we only want to make copies. We only want DNA replication to occur before a cell is going to divide. If that happens, so if we make a copy of all of our DNA, when the cell goes to divide, 1 copy goes into 1 cell, the other copy goes in the other, both cells have the exact same DNA instructions. So we only want our cells to make a copy of DNA. We only want them to go through DNA replication when they're going to divide. So right before the cell's going to divide, that's when we want our cells to go through DNA replication. All right, then we'll get to this last question. Use your DNA model to demonstrate replication. So I will say, show me DNA replication and explain what happens during each step. So you use your model here. So this is one DNA molecule. We know at the end of replication, we're going to have two identical DNA molecules. So you would explain it like this. Helicase enzymes unzip the two strands by breaking the hydrogen bonds. And then you show me that happening. So it unzips the two strands by breaking the hydrogen bonds. DNA polymerase adds new nucleotides to the old strands. So here's an old original strand, here's an old original strand. So I'm going to add new nucleotides, and this is by DNA polymerase. So DNA polymerase adds new nucleotides to the old strands. Make sure you use your base pairing rules. You can't just throw any nucleotides there. A and T always together, G and C always together. And then I'm going to do the other side too. Now, remember, true DNA replication would go all the way through. So we make a copy all the way through our whole molecule. At the very end, we wind up with two identical DNA molecules. Okay, so that's one of the most important things to know. So I'm going to do that one more time, since we haven't had a lot of chances to practice this. So I will say, show me DNA replication and explain what happens at each step. So step one, helicase unzips the two strands by breaking the hydrogen bonds. DNA polymerase adds new nucleotides to the old strands. You could say matching or complementary nucleotides, that works too. This time I'm going to keep on going, but like I said during the quiz, you do not have to go all the way and build both new strands all the way through. But I just want to make sure I cover everything here. And the actual process in a cell is way faster than what I'm moving right now. Got to deal with Google formatting here and everything. So in the end, we wind up with two identical DNA molecules. And you can actually see that, AT, 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 CG, CG. So they say the exact same things. So at this point, we've made two identical DNA molecules. Now the cell can split in two. And when it does, this one winds up in one cell. This one winds up in the other cell. And now both cells have the exact same DNA instructions. So that is what you need to know for the one-on-one quiz. Make sure you contact me if you still have any questions.